Alright, so the acid fast stain will be the next microbiology stain that we'll be talking about. Acid fast stain. Acid fast stain. Now, for this acid fast stain, you could say this is like the procedure, right? Um, but we'll talk about it when we're talking about the procedure. We'll relate it to this picture so that you guys understand it. But one thing you should understand is non-acid fast organisms. Can you see their color? It's kind of like blue. While for the acid fast organisms, yes, sometimes it could be reddish pink, sometimes it could be pink. All right. So the acid fast is actually a differential staining technique. Differential because you want to now differentiate between the non-acid fast and the acid fast type of organisms. Okay. So you are using it to identify what acid fast bacilli, like what Mycobacterium tuberculosis that can cause tuberculosis. Okay. So this thing takes advantage of the unique properties of the bacterial cell walls to be able to now what? Announce their presence. Do you understand? So for the principle, we say that acid fast bacteria, they actually what? They have a high lipid content in their cell walls, right? So that's particularly I'm talking about mycolic acids, and this will make them be able to resist what? Decolorization. If you stain it, they will not be decolorized after. Do you understand? Because the stain you actually applied was pink, alright? So after you did it, they tried to decolorize. So some other organisms decolorized back to blue, but the acid fast did not decolorize. They still maintained the stain of the color of the original stain. Why? Because their cell wall is so thick. It has a lot of lipids. So the lipid got hold of the stain and couldn't let go. You understand? So they have high lipid content in their cell walls, particularly mycolic acids, which makes them to resist what? Change to decolorization. That's after you stain it. You understand? So this property is what makes them what? Acid fast. Okay. So procedure is carbon is applied to the smear, staining all the cells, right? So application of carbon, that's the primary stain. Okay. The next one is what? Heat. Right. So the smear you are now heating it to fix it. Alright, so the smear is heated to facilitate the penetration of the what? Stain into the cell wall, or some people say fixing. Alright, so application of heat, carbon fusion, application of heat, decolorization. This one is when an acid alcohol solution is used to wash away the stain from the non-acid fast cells, right? So application of alcohol. Alcohol is a decolorizer. Can you see how the steps are? There are different ways of writing it, but it's the same thing. Then lastly, you now apply what? Methylene blue or malachite green, right? You apply to the stain, right? This one will provide a contrast. Okay. So results. Acid fast bacteria will be red or pink. Very simple. Right? Acid fast red or pink. Non-acid fast, sometimes they are blue, sometimes they are green. Do you understand? So the staining technique is essential in microbiology and pathology for diagnosing diseases like what? Tuberculosis and leprosy. All right.